Engineering virus vectors for vaccination. A viral vector vaccine uses a modified, harmless virus, the vector, to deliver the genetic instructions for a specific antigen, a molecule that triggers an immune response, from the target pathogen. Once inside your cells, the genetic material is read, the antigen protein is produced, and your immune system learns to recognize and fight the real pathogen. Common virus vectors used for vaccination. The most widely used and studied viral vectors for vaccines fall into a few main categories. 1. Adenoviruses. These are a family of DNA viruses that typically cause the common cold. They can efficiently infect a wide range of cell types, and most vectors are engineered to be replication defective, meaning they can't multiply once inside your body, which increases safety. Examples: human adenoviruses, like Ad5 or Ad26 used in vaccines like Sputnik V and the Janssen/Johnson & Johnson COVID-19 vaccine. Chimpanzee adenoviruses, CH-ADOX1 used in the Oxford-AstraZeneca COVID-19 vaccine. Using a chimpanzee virus helps overcome a potential disadvantage. Many people have pre-existing immunity to common human adenoviruses, which could reduce the vaccine's effectiveness. 2. Poxviruses. A large family of DNA viruses, most famously including the vaccinia virus, which was used in the original smallpox vaccine. Their large genome allows them to carry a significant amount of foreign genetic material. They also induce a robust immune response. Examples: modified vaccinia ankara, MVA, a highly attenuated, weakened, and replication-deficient strain of vaccinia virus. It has an excellent safety profile and has been used in various vaccine trials, including for Ebola. 3. Vesicular stomatitis virus, VSV. An RNA virus that usually affects cattle, horses, and pigs, but is often used in a modified form for human vaccines. It is a replication-competent vector, meaning it can multiply in the body, which often leads to a very strong immune response with a single dose. However, it's genetically engineered for attenuation to ensure safety. Example: the RVSV-Zbov, ERBO, vaccine against the Ebola virus. Methods of engineering virus vectors for vaccination. The process of creating a safe and effective viral vector vaccine involves sophisticated genetic engineering to give the virus three key properties: safety, the ability to express the desired antigen, and the capability to trigger a strong immune response. Steps includes: 1. Removing virulence and replication genes. The first and most critical step is to make the virus harmless. Deletion of virulence genes, genes responsible for causing disease, pathogenesis, in the original, wild-type virus are removed or inactivated. Creating replication-defective vectors. In many popular vectors, such as adenovirus, genes essential for the virus to replicate inside human cells, like the E1 region in adenovirus, are deleted. This ensures the vector can infect a cell, deliver its genetic cargo, and produce the antigen, but it cannot create new virus particles, making it safe. 2. Inserting the foreign antigen gene. The core of the vaccine is the genetic code for the antigen of the target pathogen. Construct design. Scientists synthesize a DNA cassette that contains the gene sequence for the pathogen's antigen, example the SARS-CoV-2 spike protein. A strong promoter sequence, which acts like an on switch to ensure the host cell machinery reads the gene and produces a high amount of the antigen protein. Gene insertion, using techniques like recombinant DNA technology, often involving a bacterial plasmid or special packaging cells, the antigen gene cassette is inserted into the viral vector's genome, typically replacing the deleted viral genes to make space. 3. Optimizing for immunogenicity and stability. Further engineering is done to enhance the immune response and the vector's viability. Codon optimization. The genetic code for the antigen is often modified to use the most common codons, 3-letter DNA sequences for amino acids, in human cells. This ensures the human cell's machinery is highly efficient at reading the gene and producing large amounts of the foreign protein. Structural modification. For certain antigens, like the spike protein in COVID-19 vaccines, the gene is modified to express a pre-fusion stabilized form of the protein. This forces the antigen to display the structure that is most effective at generating protective neutralizing antibodies. Reverse genetics systems. For RNA viruses like VSV, a technology called reverse genetics is used. Scientists build a copy of the viral genome as a DNA plasmid, manipulate it in a test tube, and then use that engineered DNA to rescue or generate the recombinant viral vector in a cell line. In short, engineering a viral vector is a process of stripping away the harmful parts of a virus and inserting the beneficial genetic instructions, essentially turning a pathogen into a targeted, safe, and powerful delivery agent for a vaccine.